So the goal for seeding, though, is now, obviously I didn't come up with this, rapid and uniform germination and seed emergence once the seed is in the ground. That's the goal. Differences at these stages are generally not recovered, which directly impacts crop yield. We mentioned that the seed variability across the field is tremendously different on the hilltop versus the bottom versus mid-slope. We got pH differences, we got nutrient differences. So the seed nutrient density across that landscape is also differences. One of the strategies is to try and equalize the nutrient content of all the seed, and that's where seed dressings and nutrient seed dressings and early applications of nutrients help the plant balance what isn't in the seed. So here's what happens. It takes in some water. Right here is the scutellum, and I'll show you this right in wheat here in a minute. There's a scutellum. Gibberellins are formed from that messenger RNA that was carried over that says, you know, when I get wet in the springtime, I want you to produce some hormones that are going to help me germinate. So it goes in here, and this alluron, this little membrane here, is the only live part left. It's the germ and the alluron are live. The rest of the seed, the coat and the endosperm, are dead. So there's a lot of this messenger RNA held in here and here, because here's where the alpha amylase, which is the enzyme that breaks down starch, and the proteases, which break down protein, produce glucose, and they feed it to the scutellum, which then feeds the germinating seed. Here's a simple look at that. We have a protective response in the fall, ABA dependent. In the springtime, ABA is down-regulated. GA, gibberellins, are upregulated. We get GA action, we get germination, and then we get seed growth. And it's mobilization of storage proteins, then the stored messenger RNA takes over. Just that easy. So, more hormones. Beyond ABA and GAs, the other hormones, ethylene, racinosteroids, salicylic acid, all of those hormones are also involved, and I point out a couple. Ethylene, based on ethylene, is sulfur. Prolamines are also sulfur-based. The sulfur amino acid metabolism is instrumental to physiology of seed germination, allowing the linking of housekeeping metabolic activities and hormonal regulations. You guys that have been putting down sulfur didn't even know how important sulfur was and how the depletion of sulfur across the prairies and now the northern hemisphere is influencing the ability of plants to perform. So here's my shot of the inside of a seed, and you can see here, here's the embryo, here's the coleoptile starting to move up, the radicle and roots are here. Here's the scutellum. You can see that the way the wheat plant germinates, it forms a node here in the germ on the scutellum. So here's the alluron layer, this tiny little red layer, and I'll show you a close-up of that. And here's the endosperm. So the scutellum and the alluron produce the enzymes that break down all this endosperm and the starches and proteins, and then feed it through the scutellum to the shoot. And you can see that this has already broken down some of the endosperm as it has been feeding the emergence there. And here is a shot of that alluron. This little tiny red line is in fact the live part of the wheat plant. And here's what's inside a wheat kernel. The N isn't covered. And this tells you what's important to the plant. 10% of the embryo is phosphorus, 20% is zinc, because zinc is involved in a lot of enzyme activity. Manganese is there simply because manganese has to be used to break down water for the first part of photosynthesis when the first leaf comes out. Boron is in there. And then in the endosperm, we have a lot of this stuff. But look at the sulfur. 50% of the sulfur is in the embryo. More than anything else, this is dry matter, more than anything else, sulfur is in there for helping with the management of germination. But you also can notice here that the magnesium, the zinc, the copper, and not so much the boron anymore, but these three are tied up by the phosphorus. Whereas look at the phosphorus. 70% of the phosphorus is stuck in that seed coat as phytate, and the plant uses phytases to get it, but the phosphorus ties up the magnesium, the zinc, and copper, just like it does to a degree in the soil when it's out of balance, and that sits in the seed coat and is not really utilized a whole lot, and I'm not sure why the heck the plant did that. Okay, we're going to roll through this fast now just to show you some of these images. This is barley germinating. You can see that the radical, or sorry, the coleoptile is coming out here. Here is the radical, and here are all the roots already coming out, and you can see all the root hairs are developing. Here's a pea germinating. This is corn germinating. It's the same kind of principle. This is going to be the shoot. This is the root. In soybeans, you can see what happens here. Here's a dry soybean. As soon as it starts imbibing water, you can see the beginning of the what's not called the coleoptile here, the shoot starting. The shoot comes out. The water goes in here through something called a micropyle, and this is a faba bean. And you can see here's the micropyle, and in this little diagram you can't see it well. This part is called a helium, and then there's a little tiny micropyle, and it sits right here, and that's how water gets in. And this micropyle is the remnants of where the pollen tube entered the embryo to pollinate it. That's how the plant gets a lot of its water into the hard-to-get seed. So here we have soybeans germinating, and I just wanted to show, here's the growing point right in here. And if that freezes, it's done, but that's the first couple of leaves. Here's a shot of it inside, and you can see the second leaf is already forming. This happens pretty darn early when that is emerging, and you can see that these are just coming through the ground, and that growing point is right there. So a frost late is a bugger, and in fact, actually cold rain on soybeans can inhibit this. So we usually wait a little bit longer, but here's wheat. This is the coleoptile. These are first two seminal roots, and this is the radical, which is the primary root going down. So here's wheat germinating. You can see right here, early on, you get the coleoptile moving up and your root starting, continues to elongate. And here I photograph every 3 hours for days, wheat and canola germinating. You can see that happening. And I just threw this in here to just show you the number of root hairs that are on a wheat plant, and that's what takes up the nutrients. But let's go back previous. Let's look inside of here. At this stage, about this stage, maybe 3 days after germination or water's going. You look inside of there, and there's the shoot, and here's the node. But you can see here's the node, but I want to look inside the shoot. So when you cut open that coleoptile, even after about 2.5, 3 days, the first leaf is already inside of that coleoptile and formed. See, what the mother plant has done is in the fall, it takes some of its energy late in the year and it primes the seed so that it can germinate really quickly, because that's an advantage for it. There's a close-up of that first true leaf inside the coleoptile, because the coleoptile protects the leaf. As the coleoptile moves through the ground, it doesn't open the tip until the plant is above ground. And that node, you can see right here, the node is on the germ, and this is the shoot, and it divides, and the plant feeds it equally to some degree into the shoot and into the root, because neither one can produce its own energy until the shoot has the first true leaf above ground. And here you can see the roots, the seminal roots are already out, and you can see the root hairs. And this is the scutellum. When I pull that off, this is the scutellum attached to the bud, which regulates the flow of those nutrients from the seed into the new shoot. Seminal roots start growing with root hairs, and poof, out comes the coleoptile, and there is that first true leaf coming out the tip of the coleoptile. This protects it from coming through the ground to get the shoot out. In the same time, the seed puts on 5 seminal roots that grow downward, and that's the end of stage 1 germination. So we go to leaf development. First leaf through coleoptile, first leaf unfolded, 2 leaves unfolded, and so on. So here's what it is. There's the first leaf. There's that coleoptile. Another shot. 2 leaves form. When the plant moves its internode up high, and here's where your seed is, here's the internode just below the soil surface. Now the growing point is up here. So it is now producing leaves and pumping them up here, and it's also producing the seed head right here above that growing point below ground. After it's all done, that empty seed shell is left behind, and a lot of these phytases are still stuck in that seed coat, never used. We're going on to the third leaf. We got 3 leaves. At that next point comes along between the third and fourth leaf, we get our first tiller. And the tillers all come from the crown node just below the ground. You can also have what is called a coleoptile tiller. You can see here's the crown here. Here's the first crown root. There's another crown root nub there. But you can have a coleoptile, another one that starts, sorry, not a coleoptile, a tiller that starts here at the seed called the coleoptile tiller, and that usually means that the plant isn't at a heavy enough density. The tillers move, and you can tell the tiller from the main stem because it has these little green sort of coleoptiles along the tiller to protect it as they come out, and they have this nice green vein in them so you can identify them right there. So now we have the inner crown. You can see here's the tiller coming off at the crown node, and you get the advantageous roots or the crown roots start developing from here, and these seminal roots start to move down deep, 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 and here's a crown or a coleoptile tiller starting here, just as we described. Here's another shot showing you can get these roots. So it is the crown roots that come on a little later and can move out in a broader scale relative to where the seminal roots are, which have about a 45-degree angle. These will also have about a 45-degree angle to more, but they're also starting from a higher position, so they will actually scavenge nutrients from different places than the seminal roots. Let's look at canola, and then we're done. Here we go. Germination is completed. And here you got germination in the fall time, where it's not, the abscisic acid hasn't been adequate. And for you guys that are in acid soil conditions, where likely molybdenum is short, molybdenum is involved in one of the stages of abscisic acid production called aldehyde oxidase. So if you're short of molybdenum, which may be the case where you've got pHs down in the 5 range, you may have some problems with actually inhibiting germination in the fall. You can see very quickly root hairs are put on canola. Germination takes place. The coleoptiles move up folded, so it's like taking your fist and shoving it through the ground. Takes a lot of energy. That's why canola, with its small seed, but it's almost a big chunk, 50% fat or lipids, can produce a lot of energy to push that through the ground. There it is, a close-up of it. Here is the growing point moving up in the canola plant. Before you can see it, here it is above ground where you have the coleoptiles up, and there is the first leaf after the cotyledons forming from this growing point. You can see it spreading out. So these will become 2 leaves. This will be the first leaf out, followed by the second leaf. And let's take a look. So here we have the cotyledons. So they come out uniform. Here we go. Here's the first leaf, the one I showed you just now. And here's the second leaf coming out from that same growing point. If this freezes, it'll turn black. You wait 3-4 days, that plant is dead. That's how you know. And here we have a soybean plant where we got the cotyledons moved up here, those big cotyledons I showed you earlier, the first 2 leaves, and now we're moving into development of the biomass. The last thing, here we have what is, seed treatment here, germination, nice healthy roots. One of the big risks is this. There's not much you can do after you get that darn seed in the ground. You can put on nutrients to balance it, which is the case. But once it's in the ground until it comes up, there's not much you can do. But take a look at this. This is just to show you root toxicity from varying rates of urea. Urea is kind of toxic because of the ammonia in there and the salt index. But you can see that all of these are damaged, and I'll show you close up. But the point is, look at the shoots. The shoots don't tell you that you've got a lot of root damage only by digging up your seedlings. So remember what was said earlier, if you get variable rate germination, these plants will probably not catch up and die. So just what comes out of the ground does not tell you for sure what is going to be there to actually produce your crop. And when you are out checking your seedlings, take your shovel, look at your roots. You know you should have 5 seminal roots. If you're going to see damage from fertilizer toxicity, it'll generally be at the tips of the roots because they're not protected there from the intake of these toxins. Or, you know, they're not toxins, they're just overdosage, basically. You can see right here the same thing on, and this is what you get. If you see plants like that with only 2 seminal roots and they're way above ground, you got some issues there and something to look at and forward, because you should have a whole bunch of nice healthy roots. And sometimes it's just luck. Here's urea on barley. You can see I put a granule there and a granule there, and you can see what the roots did. I put a granule further away, you can see what the roots did, a lot different. Here's MAP, monoammonium phosphate. I put one there right at the germinating root hairs. And look at the damage it does. Sometimes it's just luck in the size of the granule and where it lands. If it lands near the germ, that's not so good. If it lands on the opposite side, it's not so bad. Thank you. So that's the end of it. Take it over, Claude.
Something Taurus, I guess, holds dear to heart when we work with our partners, with our solutions, our products, is we want to ensure that what we're bringing to market and what we're selling has some backbone to it. And with Active Agri, this part of the product or solutions that we have, is we spent a lot of time and a lot of effort from 2013 to 2018 working with these solutions to ensure that what you're getting provides value and that we can show the return on investment. So from 2013 to 2018, we worked with, if you see the different parties that we've worked through on the left-hand side there, it's a long list, which is great. It covers all across Western Canada, the States, and in the actual dollar investment is a lot. So it takes a lot of dollars to make this happen. And then also, obviously, with Active Agri, we also work on some of our nitrogen management products too, and that is also a part of some of the research that we do too. So very important that I wanted to identify that we do spend a lot of effort to ensure that what we're bringing to the market has some stake to it, and in using third-party applications. We do a lot of stuff with growers on the farm, but a lot of that stuff can't be validated if just side by sides with the grower, which is good stuff to have too. But to truly have something that we can put in a booklet or put on our website is important to us. If you are interested in looking at some of our third-party data, I'm going to talk about the 2 products next, but we do have the third-party data on our website. So taurus.ag, and you go into the plant enhancement products and you can see the products. So the first product I want to talk about, we'll go to the next slide, Mike, is Active Prime. This is obviously a seed primer for seeds of all types. What's really important to notice about this product, and Mike covered off, the importance of nutrients in these products. And there's, if you look at down below on the left-hand side, you see the list of nutrients that are in their Active Prime. We didn't come to these list of products by accident. It's when you talked about the important nutrients that it takes for a seed to get out of the ground, and it was such good information on how you can't grab those 4 seeds from the middle of a head, or you can't grab those 2 seeds from the middle of your canola pod. What you're given, you do your best job to clean that seed up, but you have to, you're taking what you're given after that. So one of the best ways to ensure that those seeds have equal amount of nutrients, or very close to equal amount of nutrients, is to use a seed dressing. And seed dressings, as far as the nutrient base, if you have good base nutrients, they can help that seed go a long way. So one of the key components of our Active Prime is a really solid nutritional background to help feed that plant. If for the seeds that maybe are missing it, or the ones that aren't in that beautiful spot of that head, is want to make sure that every seed has that equal opportunity with the phosphorus, potassium, boron, manganese, and zinc. Very key components to getting a seed out of the ground. The other thing that's in our Active Prime is, get it back up there. The other thing that's into Active Prime is we have 2 modes of action. The other thing is intrinsic, and it's a biostimulant. And I guess one of the things with biostimulants, we have a really neat it's just a one-pager blog that we did on biostimulants, and it just helps you understand why biostimulants, how they work, and what they do in a plant. And what they do is they try to assist the plant to increase hormone levels or decrease them depending on what the value to the plant is. And many biostimulants can be looked at a few different ways. They do help the plant, and having a nutrient base behind them makes them that much more consistent. But with biostimulants, one of the things to always think about is how consistent are they? And most biostimulants are from an organic source, something from nature. One of the things that we pride ourselves is we actually have a patented product. Our biostimulant is patented. It's an organic patented biostimulant, so it's synthetic biostimulant. And what's neat about that, if you think about kelps and humics and other biostimulants that are in the marketplace, is that they do come from somewhere live. So the consistency of those can be variable. Yes, there's some very good ones, but there's no consistency from jug to jug. It's tough to create that. With a synthetic biostimulant, it is made in a lab. So intrinsic's been a very key component of our product, because we have a product that is made the same every time, and it comes in, we put it into our solution with our nutrient dressing. So not going to surprise you with some of the stuff that a seed prime does, but it helps provide that seed with phosphorus and zinc and manganese. It helps for very even emergence. Now you have seeds that are treated equally. Helps with the different depths, different weather events that you may see throughout germination. So you get increased development, you get the plant out of the ground faster, and you get an even emergence. And what that will come down to harvest time is you get even harvest timing or fungicide timing, neat things like that. So go to the next slide. So I guess one last thing I want to make comment on active prime is primers are, there's lots of good primers in the marketplace, and I do encourage growers. One of the things I encourage growers to do is that you only have one chance to get that seed right, and there's a lot of dollars on seed treatment. Don't forget the prime, because it's such a component aspect to make those seeds have an equal chance to perform their best. Because if you looked at back when a few of those plants that struggle to get into the ground, that yield is already lost and stuff. That you can't get that back. Active Build is our herbicide product, timing product. It also has 2 modes of action with intrinsic and the nutrient analysis. Mike will be having on our next session on April 15th. He'll be talking about what happens to that plant after it comes out of the ground, and you think of the abiotic stresses that a plant comes after. And we'll talk more about really exciting stuff. And Mike has a great way of looking at it with some great pictures of what happens to that plant once it's out of the ground and what that plant has to face. And Active Build is a great way to reduce stresses, get phosphorus and nitrogen to that plant, and other key points like zinc, copper, boron, manganese early in that season, get that plant going. Other than that, we'll look at questions. We only have, unless I'm looking at someplace wrong here, Krista, for as far as CCAs, anybody who signed up should have got an email regarding CCAs. So if you didn't get it, please, I guess, email Taurus or email one of us and ask us if you didn't get that email regarding how to either sign up on the PDF or do the download with your cell phone. But as far as one question that we had, Mike, the question I see is 5 centimal roots on wheat, how many on barley? How many in barley?
I don't know. I did those shots on barley about 10 years ago. Nobody ever asks about barley, but it kind of looked like about 5. I may have to go back to that picture. I actually don't know. I was surprised actually when I looked at that. Way back when, I didn't even know that the coleoptile comes out of one end of the barley seed and the roots come out of the other end. It's not like wheat, but I don't know. But I'll tell you what, I'll germinate some and I'll send that email out.
Perfect. Next question we got, another one popped in. Yeah, so anybody who's got questions, please send them in. We'll answer a few questions for the next 4 or 5 minutes till time is up. So Mike, appreciate the answer that you're going to find on that. So, but the next question is, how calcium would play in germination? What does it do? What does calcium do? What's its responsibility?
Well, I didn't mention that, but I'm glad you asked the question. One of the prime uses of calcium is that it's one of the ions in the actually alpha amylase enzyme along with chlorine and a bunch of sugars. So it's key to the functioning of amylases to break down the starch. It's also key in terms of building the new cell walls, because it takes calcium along with that boron. So you notice that in the seed itself, it had quite a bit of boron. And boron is used for a lot of stuff, but primarily I look at boron and potassium in terms of building the root cells and the shoot cells. In the root cells, you got potassium and boron and calcium, because calcium is used to build a cell wall, boron to sort of stick it together, and then the potassium with water expands the cells to drive the cells longitudinally and force the roots through the ground, but it also grows the cells longitudinally and forces the shoot upward. So if you want strong shoot growth and you want alpha amylase to be functioning for you, it looks as though you need a fair amount of calcium in the seed. It's going to pick up calcium too right from the soil, but you know, I didn't mention that when it's really cold, the roots aren't growing much. Things like phosphorus aren't being mineralized by microbes. So those sort of seed dressings provide those tiny, tiny roots with immediate access to those nutrients like zinc, boron, and so on during those cold periods. And once the roots get going, then it can sort of access more. But it's a combination of all of those things, but calcium is key. The other big role of calcium generally in a plant is it's picked up largely right behind the growing root tip, and then it's not picked up as easily further up the root. It acts as a real signaling agent in plants of all kinds, the actual levels of calcium, because it's a signal on what the root is finding when it moves through the ground.
Thanks, Mike. We got another question. Do you know the optimum levels of nutrients to have in a wheat seed? And next part of that would be, would you say you can use that table, you know, as a guideline, the table that you showed?
Actually, we don't know what the amount should be, and I know that A&L Labs is actually developing a database on that. We haven't, you know, we started on it, and I'll go back. I used to do a lot of work with ATP, and ATP, I started trying to do that and looking at nutrient density in conjunction when Dr. Chakmak was around, because we were looking at fortifying wheat plants with zinc and applying zinc late in the sort of the milk stage to get the levels up. So we started on it, and then we basically after 2 years kind of quit. And I was talking to Greg Pattison, and he is trying to develop that. I'm telling A&L that one of the things I would like to see is to, in fact, develop that database for us in Western Canada. So the answer is no, we don't know what the maximum or the ideal should be. On the other component of that, I think that table of what's in the seed is a good guideline to tell you what nutrients are important in germination, because that's what the plant is trying to get. Now, the difficulty we have is that if you haven't got phosphorus for the mother plant and you haven't got zinc for the mother plant and you don't have water late in the season to help it move those, you know, you're going to have difficulty getting what the mother would want into that plant. She's going to do the best she can, and that's why those two seeds would be ideal. And if I'm going to do this, which I'm tempted to do it, because I think we have to get around to that nutrient density to complete the whole cycle. We do the soil test, we do the tissue test, and then we forget about what's in the seed, because that would tell us what nutrients are difficult to get and what nutrients the plant can't get. So by putting on, you know, a seed dressing and then taking a tissue test on the early germination and taking a look at what's lacking, we can then try and fix it with a foliar treatment on the micros and allow the roots to get going to go into the soil zone and extract nutrients as best it can. And if you've all heard me, canola is one of the best plants at doing all of those things.
Thanks, Mike. One of the things I'll find interesting as we look into that is how closely those nutrients match the removal rates, which I think will be interesting. That we expect. And then also I would like, you know, where we see our best seeds, you know, you talk about that, those middle rows there versus the rows that are on the upper end or bottom end, what the nutrient levels within what we'd call our best seed or our best germination seed versus the ones that aren't. That'll also be interesting to see what the difference in values of those would be too.
I think we could get some real clues if, you know, and the technology must be there to do individual nutrient counts on individual seeds. It's going to cost some money to do that, and of course it's going to be tremendously variable. But if we compare the seeds, we will get some clues on which ones are the most important ones, I think.
And it's no different when you apply a primer, a nutrient primer, is that you see, you know, and with and without, you'll see those ones without will perform very well, but then when you have the primer on, you'll see them all perform equally as well. So it'd be interesting to match how those ones that are, you know, performing on their own with or without the primer, and the ones that really take advantage of the primer.
Lots to do.
Next question. Just have a couple more questions. We'll try a couple more, and then we'll shut her down here at a few minutes after. Next one is the number of settlement roots determined by the nutrition in the soil?
No. It's genetically programmed by the plant, and it has no more, and it shouldn't have any less. If the plant is, if it hasn't been killed off or diseased or whatever.
Okay, so that kind of answers the second part of the question. Is there any other reasons why a plant would have less besides fertilizer burn and pests? So it should be the same regardless.
It should be the same. Just like, you know, wheat has three anthers, canola has so much. These are all programmed genetically in the plant.
A couple questions here. With the seed treatment, I'm thinking this is maybe the seed primer. How long after treatment can the seed be stored prior to seeding? Maybe that's a two-part question. Is how long does it have to be stored? For the most part, as long as you give it a couple hours of drying time is fine. As far as the active prime, I've seen guys even use on-the-go treaters and be successful with that when they're applying the right rate. So when you truly do get the right rate of your seed treatment and your nutrient dressing, it does dry very quickly. That's kind of the one neat advantages I didn't get into with the active prime, is it's a very easy product to work with and dries relatively quickly. So it's next one is with active build in cold spring soils coupled with damp conditions, what time application would you suggest? This is a conversation regarding active build. When would I best use it? The best timing I've seen, the best responses I've seen is if things are cold, is the earlier the better. Trying to get those, and Mike, I'll let you comment after. This is just from my, you know, working with the product for the last 6 years, is when you get in there early when things are rough, it really helps get that plant growing roots, because the plant really tends to want to stall, and if roots aren't moving, therefore you're having a lot harder time getting nutrients. And when you get that the intrinsic and the nutrients into that plant, it really makes that plant, and we've even seen it where we've had a plant stalled out and we apply build to it, and we can get those roots moving down using some of those new weather stations and root growing stations that that are in the marketplace. Any comments on that, Mike?
Yeah, for sure, because you know, once it warms up, everything speeds up. So both the mineralization and the availability of nutrients provided by microbes and the exudates in the roots, as well as the speed of growth of the roots, just allows the plant to access the nutrients a lot faster and more efficiency. Is during that early cold stage that it's really tough. And I did, and because I just kind of joined Taurus, I did a lot of work long ago in terms of looking at sort of seed primers, and it was easy to find tremendous advantages in root growth. And I measured root lengths using some equipment I have here at home, and it was just clear as all daylight that those roots just took off with that little bit of, it sort of surprised me because it seems like so little, but yet, you know, it doesn't take much. That seedling is just tiny, so it doesn't take a lot to really enhance the growth.